All right, quick review. We talked Friday about the components of the endomembrane system, which was this kind of long continuous path that certain materials take through the cell. The general pathway we said was DNA, and then RNA reads the DNA and takes that information to the ribosomes. The ribosomes stick to the ER and dump their proteins into the ER. The ER sends little vesicles to the Golgi apparatus. Vesicle is a word we did not really address on Friday. Vesicle is kind of the general term in the endomembrane system for when you wrap a membrane around something and send it somewhere. That's a vesicle. In the case of sending ER to Golgi, we call it a transport vesicle. In the case of sending something from Golgi to outside the cell, we call it a secretory vesicle, as in it's secreting something from the cell. We also have incoming vesicles, which is where we run into things like lysosomes, right, which break things down, and vacuoles, which store things. But so basically, DNA to RNA, to ribosome, to ER, to Golgi, to wherever is the general pathway. Side trips include the vacuole, which is for storage, and a lysosome, which is for breakdown of old cell parts or waste, or in some cases, other things. Questions about that before we move on? That was a big chunk. Big functional chunk of the cell's kind of machinery. Anything that's not clear there? Okay, I'll trust you. All right, so a couple organelles left here. Oh, and also you started drawing a dream house, right? Which is due tomorrow. I hope that's going well. Finally. The mitochondria. The mitochondria is weird. It's a weird structure. It is the organelle that primarily releases the energy from your food, specifically glucose, that you break your food down into, right? You don't just send food into your cell. You send food down your digestive system. Your digestive system busts it into a million little pieces, and the most functionally useful of those pieces is glucose. And glucose goes straight into the cell through a glucose channel, remember channel proteins, and into your mitochondria. And your mitochondria does a thing called cell respiration, where it takes a series of breakdown steps. It breaks glucose down eventually into carbon dioxide. And in the process makes a whole bunch of a chemical called ATP. That's where we're going next, by the way. Chapters 8 and 9 are all about photosynthesis and cell respiration. So you get the big picture of this then. For now, we're going to talk about how the mitochondria is weird. First, it's weird because there is a double membrane. It is one of the few double membrane organelles. Second, it's got like all its own stuff. It's got its own little loop of DNA. It has its own ribosomes. In other words, it's making its own proteins independently of the cell it's in. It even, like every other organelle, we talked about how everything's made of membrane, and the smooth ER is always making new membranes for all these parts. And if you want to divide, you're going to make more ER, and you're going to make more Golgi, and you're going to make more lysosomes. Not the mitochondria. Mitochondria is going to go ahead and make more of itself. It literally reproduces on its own. It's like a little stowaway cell. It's like a cell inside your cell. It behaves very much like that, which is weird. Last weird thing, and it is super weird. All your mitochondria are descended from your first mitochondria in the first cell that was you. And the mitochondria in that cell, the zygote that became you, none of them came from your dad. inherited entirely from your mother. Now, see if you can figure that one out. Why would that be? Why would you only get mitochondria from your mom? Anybody think of a reason?
Because the food and nutrition that you got from your mom when you were in the embryonic sac only came from your mom. So
but this is pre that. Like, this is the first cell. This is your dad's sperm with your mom's egg, and they fused to make one cell, but somehow dad's mitochondria didn't make it in there. Only mom's did. Why would that be? We're about to learn, tomorrow we'll start learning about how cells only kind of have what they need. Like the coloring book picture is a lie. It's a very useful lie to teach you all the parts, but cells don't look like that. Cells have a whole bunch of whatever organelles they need and not much of the others. What do you think a sperm needs a whole bunch of organelle-wise? What does a sperm do all day? What does a sperm do all day? Were you not in class on Wednesday? Did I not do my interpretive dance for you? Cilia, flagella. What does a sperm have? Flagella. What does it do all day? Thank you. What would that require a lot of? And which organelle is the powerhouse of the cell? Oh my God, you're looking at it. So why didn't any of those make it into you? Why didn't any of dad's many mitochondria make it into you? Oh, come on. You got this. Amanda?
they died.
No, they didn't die. Where were they all? Where would all the mitochondria in a sperm cell be? In the tail. Tail doesn't make it in the final product.
Wow.
Yeah, crazy, right? So that's why, and this is really cool because the mitochondrial DNA doesn't change very fast. So like you can trace mitochondrial DNA back through the female line of any eukaryote, like really far back. Like we talk about mitochondrial Eve, like you can go all the way back and track all these mutations. It's super cool stuff. Anyway, that's the mitochondria. Then we got stuff that's specific to plant cells. Do you know what that organelle you're looking at right there is? Good. It's called the chloroplast. Mitochondria is the organelle that breaks glucose down. Chloroplast is the organelle that makes glucose. This is where photosynthesis happens. And again, that's chapter 8. That's where we're going next. Not going to go through all the details right now, but it's a lot. What we're going to do is look at how weird they are. Chloroplasts have a double membrane. Chloroplasts have stacks of photosynthetic membranes where photosynthesis happens, but also, just like mitochondria, their own DNA, their own ribosomes, and they reproduce independently of the cell. Again, they're like little stowaway cells, just kind of along for the ride, but doing lots of good work, right? So real similar, the way that mitochondria and chloroplasts are weird. And we're about to talk about what that similarity means. But first, the other thing plant cells have that animal cells don't, which is cell wall. And that's all about rigidity, right? Because plants have that weird body plan where they got to have the leaves high and the roots low and as little as possible in between so they can be as tall as possible. So they got to stay rigid. In plants, the cell wall is made of cellulose. Fungi have cell walls made of something else. Bacteria have cell walls made of something else. But in plants, it's the polysaccharide cellulose. You find the cell wall just outside the cell membrane. And so this helps keep the plants rigid, but it also, of course, comes at a cost of movement. You don't see plants walking around. Okay. So now we come to the part where a scientist named Lynn Margulis is thinking about how weird mitochondria and chloroplasts are and wondering what that common thread means. What does it mean that mitochondria and chloroplasts both have their own DNA, their own ribosomes, reproduce independently in the cell? Like, how did that happen? What does that look like in terms of the evolution of eukaryotic cells? How did it work? And I think what kind of tips her off is she sees a single-celled organism we've mentioned before, an amoeba. Remember talking about amoebas? Amoebas, how they're just this big, sometimes they're a whole millimeter in diameter. And they make extensive use of their cytoskeleton, their microfilaments in particular, to change the shape of their cell membrane, right? They got all these kind of interwoven threads that they can shorten and lengthen and actually change their shape. And that's how they move. They kind of ooze around by moving their cytoplasm that way. But it's also how they eat. So like, for example, if an amoeba comes up against something it wants to eat on the outside of it, it doesn't have a mouth, but what it does is it starts to change the shape of its cell membrane and kind of bend it around the thing it wants to eat. See what I'm saying? Like it starts to actually kind of envelop the thing. Think of it 3D, right? It's not 2D, but that's how the only way I can draw because I'm not a good artist. But like, and eventually, the thing about the cell membrane is it's this fluid structure. It can pinch off if it needs to, right? And so eventually it does. It pinches off the membrane, and suddenly the thing it wanted is inside it. Yeah? You get it? You can picture it? Although, technically, you could argue that it's not inside it because there's a piece of the outside right here. But that's just a matter of now getting rid of this membrane, right? If you want to digest that as food, which the amoeba does. Well, that's kind of the aha moment for Lynn Margulis and the endosymbiotic theory. Margulis observes the weird things about mitochondria and chloroplasts, the double membranes, the DNA. And also she observes it's not like your DNA or my DNA. It really looks like bacterial DNA. It's circular. It's a loop of DNA that in a bacterium we call a plasmid. And in fact, now that we know how to sequence DNA, it has been supported that mitochondrial DNA is basically bacterial DNA, directly related to ancient bacterial DNA. Even the ribosomes, they're not like your ribosomes. They're smaller. And their RNA sequence looks like bacterial ribosomal RNA. Even the division, even the way mitochondria reproduce looks like what we call binary fission in bacteria. It's simpler than mitosis that your cells do. And so after the kind of aha moment of, whoa, look at how amoeba eat, and knowing that these mitochondria and chloroplasts look like little bacteria living inside cells, she goes, oh, back up. Now it's not just a chunk of food, inanimate food that the amoeba is engulfing. Now it's a bacterium. Now let's say it was a little bacterium that the amoeba wanted to eat with a little loop of its own DNA and its own ribosomes and the ability to reproduce. What does it look like when that amoeba is done internalizing, endocytosing? That's the process, endocytosis. What does that bacterium look like inside there once the amoeba does that? What does it have that chloroplasts and mitochondria have? Well, yeah, it's got its own DNA. It's got its own ribosomes. But the other thing, the thing that is explained by this process of wrapping your membrane around the thing you're internalizing. You see where it's going? Complete the process. What do you see? Amanda?
Double membrane.
Double membrane. Chloroplasts and mitochondria both have a double membrane. Imagine this, amoeba, which is really good at like moving around and getting food, accidentally engulfs a little critter that's really good at digesting it. That's really good if you feed it glucose. This little bacteria right here produces mad amounts of energy. And so by some twist, the amoeba fails to digest this thing that it's internalized and just leaves it there. And suddenly both of them have huge advantages. Suddenly the amoeba is getting energy like crazy. Suddenly the little bacterium is getting more food than it ever had in its life. They both thrive. All it takes is for them to do a little better than their neighbors and to pass on this little symbiotic relationship. And suddenly we have an evolutionary trend, right? So that's the idea. It began as free-living prokaryotes. Either it was engulfing, or maybe the bacteria was getting in there, trying to parasitize the big cell. Either way, all it takes is that it benefits both of them, and it's a relationship that lives on. And we're not talking about a time in evolutionary history when there were, big critters walking around. We're talking like single-celled life only. And so all there are big cells and little cells. And some of those big cells are getting their food by engulfing things. Sorry, Nick. And, all it takes is one little cell to be engulfed by one big cell and for them both to benefit from the relationship, and it's going to get passed on. Questions? So that is something that we used to teach as the endosymbiotic hypothesis. We now call it the endosymbiotic theory because it has been so well supported. Again, through DNA sequencing and RNA sequencing, you can basically just trace mitochondria and chloroplasts back to ancient bacteria. They get their lineage directly from them. Okay, that is good for a Monday. I'm not going to give you more homework. You've got your cell, not cell. Who said cell? I didn't say cell. Your dream house to complete by tomorrow. All right.